Okay. So, as you know, the anti-cancer agents do have their own problem. So the clinical trial, from the day one, it's getting improved and getting better. But we are learning from each clinical trial. But I wouldn't call that as a bad clinical trial design. Rather, and then the cancer drug again, they are not bad anti-cancer agents. It's a combination of both is what is making the future. So therefore, I wouldn't agree with that statement.
So thank you. So that you know that 2022-24, the World Cancer Day slogan is close the care gap. Next. So how to close the gap? So priorities from a cancer research in the low and middle income countries. So this was a recently published paper led by Dr. Pramesh. So it showed that only countries like where Dr. Vivek and Dr. Mani are there. So the cancer research currently is heavily skewed toward the high income countries. So what do you think?
So basically, in this perspective, I will just say we have just now been asked to apply for the international grant at least for funding for some research. The amount goes up to 5 lakh rupees. So they were asking us that you try to develop some concept that what you are going to do in research. As I said, if we want to go ahead with research in our sort of population, we will focus mostly on the genomic research, molecular oncology. Now, when we think about molecular oncology and gene sequencing, there lies the most important fact of what is going to happen. So I think that is the most important thing to consider in our system.
So there are two sides of every story, and that usually lies somewhere in the middle. Next. So hype and hope. So cancer treatment should benefit patients, a common sense revolution in oncology by Vishal Goyal. So Dr. Vivek, so what is your take on this? So we talk about game-changing treatments, targets, but it doesn't reach a common man, and the benefit is not huge.
So I guess it's a
yeah, so Dr. Vivek and Viren sounds very similar. So I was wondering, it was a question directed to Dr. Vivek, but yes. So what I would suggest is, since we have traveled 5 decades from histopathology-based morphological diagnostics to immunohistochemistry, and now slowly moving into gene-based and NGS-based gene panels. So we are kind of decreasing the cost and increasing the numbers. The only way going forward is to join hands. As many hands we join, we decrease the cost. So in a country like India, where when COVID came, we were doing tests at 3,500, 4,000 rupees for our RT-PCR test. But I'm proudly telling the place where we are sitting, Odisha, we have shown the way. We have decreased the cost of RT-PCR testing to 300 rupees. So if that is possible, cancer treatment in next 2 years, 3 years will also go down, and research over the way. We are confident in that.
So Dr. Vivek, so as Dhiren said, so bridging the current with the future. So this is a recent publication last month. So we have been, as a clinicians over the country, we are doing a comprehensive genomic profiling. So 12 of my colleagues almost collected 2.5 to 250 around these reports, and they just analyzed. It was led by Dr. Ajit Mathur. So clinical benefit of comprehensive genomic profile for advanced cancer in India. So let me come to the conclusion. We observe that in a low middle economic country setting, 10% of the patients receive targeted therapy on the basis of this report, which starts from 80,000 to 3.5 lakh rupees. If I talk about the Rose Foundation, so only 4% of the patients who underwent this test actually got a clinical benefit. So what is your take for a country like us? I'm not talking about India or Texas at this moment. What are they?
Yeah, I had so much of echo. I couldn't hear the question properly. Can Can
So
someone?
I just would like to summarize. This question will be for both Dr. Vivek and Dr. Mani, because your DNA is made in India. So therefore, as fellow Indians, what's your take of how to get that 4% patients receiving high-end genomics-led therapy to make it 40%? How do we go about it? Your take. I know it's $1 million question, but your thought process going forward.
And I think, let me tell you, thing is that, when we started the sequencing here in the US, maybe 2 decades ago, it was the same thing, right? Even now, right, only a few handful of cancer centers are testing it. I think India can provide the way of the future by getting the costs of sequencing down. Again, still in the US, the cost of sequencing is $3,000 to $5,000, and we're having insurance to pay for those. And I'm sure, if India, everyone here can put their minds together, you can get it down to less than $20, right? It was possible for COVID. You can make it, and then sequence, offer sequencing for the entire world at a fraction of a cost, and then make it a part of diagnosis. That is number one, right? So India can be the leader for genomic sequencing for the world. So that is number one, right? Number two is genomics not just provides opportunities for treatment, right? It also provides opportunities who not to treat, right? For instance, if you sequence a colorectal cancer genome, if you find a KRAS, you're not going to give cetuximab to the patient, right? So again, it would also tell you not to sequence it. And again, most of the sequencing is done in patients in whom the cancer is relapsed and refractory, when the patients are in terminal or in a hospice-like situation. So again, you can say that, hey, genomically matched therapy is challenging in patients who are in a hospice terminal situation. We need, most of the genomics and targeted therapies work efficiently when the cancer is simple, earlier in the disease course, earlier at the diagnosis. And again, to me, it's a paradox on how even the subsets of patients respond at the end of life. So I think, more and more, again, the reports, earlier reports are very encouraging. In fact, it should motivate people to do more and more testing in the earlier disease course. And one more thing is also identify the genomic differences between the Western world and Indian world. I think we cannot just give up, right? If we do the same thing over and over again and expect different results, we might not lead to where we started from. And again, everything has to be done in parallel. I think, of course, genomics has to be done. Basic cancer tests should be administered. We cannot wait for malaria to be cured before we start cancer treatment. Again, we need to do everything possible as oncologists, scientists, and physicians to keep the needle moving forward. I think India can definitely lead the way. There are so many countries. As I showed you in the map, only a few countries have precision oncology programs. If the cost of genomics is down, and India and you all have a solution for that, right? Every genome can be sequenced in India.
So thanks. So since we are running short of time, I have to rush to begin. So this is the first case of Dr. Sevanti. So just an example, I'm just going through the slides. So this is a metastatic pancreatic adenocarcinoma based on calcium and that or a broad-based GS. So none of this was positive. So KRAS G12 became positive. And we talked about the 70 also. So we chose chemically by the Umas secondary. Next. So this is a metastatic one adenocarcinoma. So these are some examples. So based on 10 carbo, bevacizumab or 10 carbo, you can see or TMG of 9. So again, this was the patient of KRAS G12G, and probably there will be evidence of genetic and specific. Next. So another unknown primary or failed on Pathlox and Caplox with bevacizumab, recurrent ascites PS2. You can see a high TMG. So these are all these mutations detected. So we thought of starting this patient on an immunotherapy by this the patient had a cardiac event, and we could not start. Next. So three things that change the oncology world. So we know the three apples that change the world. So Dr. Mani, so A, B, C, just a quick buzzwords. Three things that change the world. Oncology world. Go back to the slide.
I think the the sequencing really made a huge impact, and just going to make even more further impact. And you put an apple there, and the technology really revolutionizing the world. Information, the data. I think I would say give a highest importance to data, because one of the things which I was going to say earlier to the previous question was that the reason why MD Anderson is number one is because besides giving a perfect care, etc., the patients who come to MD Anderson are most of the time are terminally ill. If you notice in the same thing in India, patients don't go to see an oncologist until the cancer is in a terminal stage. So therefore, there's a lot can be done. A lot of data could be generated in Indian population, which could set as an example for the world, as Dr. Vivek Subbiah said earlier. So I think the data is the key, and India can generate a lot of data which could really change the world. Yeah.
So at this moment, so we are also struggling with those patients who have a medical insurance. So if we have they find that a hypertension with a lung cancer, so the insurance bills are rejected. So that's why I put a code. So if somebody has a fall and broken his limb, so that since gravity is a pre-existing condition, so you might not get an insurance. Next. So how do you decide? So Professor Somya Panda is there. So you are wanted to do a broad-based exchange. So how do you decide?
So we look at the targeted medicines. So we should not do an exchange which doesn't help you in getting the patients and rather causes anxiety. So before sending the report, you must clear in your mind what is the target that you are looking for, what is the therapeutic potential of that particular report. And so therapeutics should not, because therapeutics is the diagnosis. In the diagnostics, it is the therapy followed by diagnosis. So interpretation of the report, the availability in the local place, and whether or not the financial part, multiple things have to be taken into account.
So now, so once the report is there, how do you back? So these are the variants which you need to see. Next. So the takeaways next. So cure is an erroneous target. Cancer prevented is better than a cancer cure. Next. So when you hear who beats the thing of horses, not zebras. So we are in India in nature. So it is easy to get a horse than a zebra. Next. So challenges and unmet needs are huge because there is a tsunami of informations. Next. And so in this era, we are moving from disease-centric to a state-centric, and today we talk on World DNA Day about gene-centric. So at the end, we are continuing this protein related to modernity. Next. So thank you, Dr. Vivek and Dr. Mani, all the way joining. So have a blessed day. So you have to rush to your clinic. Thanks for joining and sharing. Thank you so much.
It's really a pleasure having Dr. Mani, a friend of mine, and my Ramchandra, ex-alumni, Dr. Vivek Subbiah. It's really, we are all proud of you. You are doing both of you are superstars in Texas. We hear a lot, we follow you. And ILON is a great venture. We have a lot of good collaborations in the future on this very World DNA Day. Thanks for your support, and really appreciate your time. Thank you so much. Have a great day.
Thank you. Thanks again. And I think the ILON is going to is in the process of partnering with inDNA with the Dr. Byron Banerjee. So you will hear more from him about ILON from Dr. Banerjee. So thank you, everyone. Thank you for hosting us.
Yeah, once again, thank you so much. And I see, every time I give a talk in India, there's so much enthusiasm, there's so much energy there. Again, as I showed the world map, in the next decade, I hope that will be flipped completely, right? I think India is, it is a future. All right. Congratulations and thank you so much for having me on. Thank you. Good luck.
Bye-bye. Thank you.
So thank you all the delegates and our esteemed panelists for making this session so interactive and informative. So now I would like to invite our honorable chief guest, Professor G.K. Dutt, founder director of National Cancer Institute, Delhi, and our guest of honor, Professor Dr. Malatendu Sarangi, the director of AHPIC, Acharya Maria Postgraduate Institute of Cancer on the dais. Now I request Dr. Banerjee to welcome our chief guest, Professor G.K. Dutt on behalf of India.
So it's again it's a great pleasure, and I am very small person to honor a giant, Professor G.K. Dutt, and it's really a blessing that he has graced this occasion and made this World DNA Day so special for all of us. So on behalf of the entire team of World DNA organizing committee, the inDNA fraternity, and particularly he has just put his director's cap down, but he as a cancer worker, as a cancer man of this country has not stopped yet. So from this land, from Bhubaneswar, from all of us, from Odisha Society of Oncology, I request Professor Sarangi sir to honor Dr. G.K. Dutt on this inDNA forum.
Now I request Dr. Biren Banerjee to felicitate our guest of honor, Dr. Malatendu Sarangi.
So I request Professor Madhavanandakar to come on stage, and Professor Sarangi and Professor Kar will chair this DNA lecture session by Professor G.K. Dutt. So, please. So Professor G.K. Dutt has completed 5 decades, 50 years in his career. He has published more than 250 papers. He has cited 25 PhD scholars, and he has changed the length and breadth of cancer demographic of this country. So he is going to tell you about what cancer is, current status, and how cancer is going to look in the future. He likes cancer because he is saying cancer is a fantastic disease. Why? Just listen to him.
Thank you, Dr. Banerjee, ladies and gentlemen. It's an honor on my part to be right here. In fact, as a guest, I am part of this stage, and I always feel to come back and enjoy this place. Well, after you have heard all this, I would like to tell in brief what we have all done in this country so far. And at the end of my talk, you realize that we have achieved a lot. And I'm sure my life stay will be fulfilled if I finish cancer during my lifetime. Why I am telling so, I will give you proof of what we have done in this country. All of us. All of us have done in this country. We have reduced the incidence of cancer. We have cured cancer. I'll show you all. Nobody listens to you without a proof nowadays. Your own children don't listen. You don't give it. I'll tell how cancer is a preventable disease, how cancer is an early diagnosis disease, and how cancer is a curable disease. That's why I always say cancer is a fantastic disease. If God asked me what is the best type of disease today to have for a doctor, for a patient, and for a relation, I'll say cancer. The reason is so much can be done. You tell us in so many years of our life what has changed in diabetes. Is it preventable? How can you prevent it? Common disease what we are facing today, everybody, every family, we have diabetes, we have hypertension, we have thyroid, we have cancer. You cannot, how do you prevent it? Can you take blood every day? If you are not right, it will become bad. Diabetes is not cured. Similarly, hypertension is like a molecule. You can certainly control it, maintain it, but life is not mathematics. You cannot come every day, get up in the morning and check your blood pressure, take an every day, or 2 hours late, you are, upset. So in that context, something about diabetes. Cancer is 50% preventable, 50% preventable, and 50%, curable. And I'll show you that. Why I'm talking about translation research today, I'll talk about the country, what has been done by all of us. When we got independence, lifespan was 32 years. Today is 70. It has happened only because of science, science, and science. In Odisha, when I was studying here 50 years back, there was, water lesson, there was electricity lesson. We have surplus. This has all happened because of science. Incidence of cancer is coming down, the cure rate of cancer is going up. And we have published it. I'll show you the published material from this country. This has all happened because of science. So I think what we need to do more is to how to finish the cancer altogether. And next one, please. Can somebody help me with it? Well, I advised the Government of India on Cancer Affairs, so I got a hand onto some of the cancer slides. This was a picture of 1931 in our country. 1931. If you see the causes of British India, cancer is big figure in the support. We were dying, not much has changed, since our independence also. Till 31 till independence, this was, when I was a student in the Medical College, whole village used to go back down because of Talga. Now we are not listening to Talga. How are we going back? So that is the issue. Well, these were they used to call it radium therapy center. This was the situation in 1931. And there were 8. And we will be happy to know that General Hospital Cuttack was one of them. 8 centers and one has got 7 in India. Only 8 centers were there. Because cancer was not an issue those days. And we all know next one. Yeah. With the control of the communicable diseases, non-communicable diseases are the biggest killer, and we better have to deal with them. That's one. Well, this is our own statistics. Again, these are all statistics which are there in the ICMR website, NCDIR, National Council of Disease Informatics and Research, based in Bangalore. It's ICMR. This is the figure which has been published by Dr. Prasad Mathur, the director of NCDIR. This is our figure. 14 lakhs new cancer cases. Again, this 350, 50% preventable, 50% early diagnosis, 50% curable. Is it not a fantastic situation? We want to make it 100%. That's different. But 50% diabetes is 100% not preventable, 100% not curable. Well, India is an epidemiological paradise for cancer. India is the only country in the world where the lowest incidence of cancer is in a place called Barsi and Shuldi regions. It's a Maharashtra, village in Maharashtra, where the incidence is 40 per 100,000. 2, Highest incidence is 270 per 100,000 in a place for IT or the capital of the world. Nowhere in the world. If you see in US, it's around 450 per 100,000. UK, it's around 350. Asia, 250 to 300. But India, if somebody asks what is the incidence of cancer in India, that's no Indian incidence. Because India, according to Cancer United States of India, 40 to 100, 40 to 280, and you can go through the report of ICM. Refer to the report of ICM. But this data is continuously being published since 1982. Similar, 7 times differences in the same country. Why? I'll show you. One of the reasons, like for example, we people, you know that the place of Guwahati, the capital of Assam. Actually, if you see in the Google, it's Guwahati. Okay, it's called Guwahati. Gua means Gua, Subari, and Hati. Gua ka hai, so cancer wani ho to kaha. The place is called Guwahati. You go to a place again in India, like Punjab, Sardar do not smoke. Because they can't smoke, we don't do that. Sardar don't take part, because we don't become regret. In Punjab, I have not seen in my 50 years career against no Sardar ever had a audience. The white people, they smoke, like Marlboro Man, when McLaren had, lung cancer, court case, and all that, they lost. But they don't chew pot, so they don't have oral cancer. So the cause of oral cancer in India, why can't we prevent it? So the cause is here in India. Well, if you see the tobacco-related cancers, eminently preventable cancer, I'll show you again about data. Out of 17 tobacco-related cancers, 7 are showing a negative rate. The other cancers are infection-related cancers, cervix and hepatocellular, cervix because of human papillomavirus and hepatocellular all are controllable by vaccines. I'm sure the girls sitting in this room, 90% are not vaccinated. This is no excuse. And what has happened in this country, 50 years back, the incidence of cervical cancer was 45 per 100,000 in this country. Again, go through this. Today it's 10 per 100,000. Kerala is 7 per 100,000. So, like Australia, which is going to finish cervical cancer next year, I'm sure in another 10 years, we will also finish cervical cancer. So, cervical, definitely, it should be tobacco-related cancer, no tobacco cancer. So, 50% cancer are gone. So, load from 14 lakhs will become 7 lakhs. So, we will be treating 7 lakh cancers. So, big cancer centers, the number will become reduced. How it can happen? By smart research, from base to base. And what we're doing, that also has fantastic things are being done in this state like this inDNA, which has been done. This is again the reference. The biggest tragedy is, sorry. Yeah, okay. The biggest tragedy is Asia has the maximum population, maximum cases, and maximum mortality compared to US, which are 13% of population but 14% of mortality. This is the situation. And Europe again. So our problem is here. Can you please move this slide? It's again technology problem. Next one. Well, this is again another piece of literature saying that in India, cancer got a moved trail that figure what means for unit population. We are adding on Australia per year. That's our issue. That's not a scientific issue. But in that way, burden will increase. But the incidence will come down. So next one. Well, this is about the distribution of comprehensive cancer center. You'll be surprised that the maximum distribution of comprehensive cancer center as it is South and West. East, South, and the North East of the list. If you see the next one. See the number of radiotherapy centers. I have basically radiation oncology. Radiation oncology is the negative factor. It's not very important. It's equal as cervical, medical, research, everything. But you can't have a Jew with just a monkey. You require a otherwise nobody will go there. So this is just a, it's not a white to us. So number of radiotherapy centers in India, you see, again concentrated to breast cancer. But that's gradually changing with approaches of new government of India policies. Next. Oncology workforce again cervical medical variation. There are very limited oncology workforce and that's increasing rate. Next. What is the tobacco problem in India? That's the most important tobacco. Tobacco is the major cause. 3,500 tobacco related deaths every day. And in India, and 40 to 50% are tobacco related. This is the source. And actually, last night, cancer, there is a branch of ICU no, that's okay. 90% of cancer because of tobacco related cancers. We can prevent that. This is another way. I don't know if one of you has gone to a house and all these pills. Assam is playing. Most of the Assam is playing. To go to other 7 states where most of the areas in the hills, this is how they live. This is the house. I am taking this photograph from a place called Giro in Arunachal. I found that place. And this is their house, not the meat, the whole piece of animal. This is animal is hanging from the top. Fire is burning 24/7. So this meat is being smoked, being charcoal, and people are smoking everything. So all the 5 ingredients of happens. And this is with the educated, intelligent ones. It's like Arabs, who, even though they have money, once a year, they will go here, air conditioned tanks to the, to their rooms where they came from. So this is the habits I have. This is what not this. That is the reason why incidence of cancer is so high. And we have got that. Next one. Well, this is another piece of literature, very important. Suppose government asks us, where is the evidence that cancer is a big killer? This is evidence. This is a study, it's called prospective urban rural. This for our students in this audience. In pure study from McMaster's Canada, where it proved for the first time, documented for the first time, that in HMICs and many of the NMICs, death from cancer has overtaken cardiac as the major cause of death. And what we tell India is that HMIC, I think it's a so we are facing already this problem on adequately. This is the slide to tell our policymakers that cancer has overtaken cardiac as the major cause of death. A scientific problem. So this is what you want to see. Next one. This is another great topic. Next one. And go step by step. See, this publication again says that 8 million people die each year of cancer. Okay. 1 out of 6 deaths globally. Exceeding number of deaths from HIV/AIDS, malaria, tuberculosis together. Again, another dangerous side, another literature. And the cancer incidence is estimated to double by 2035. Another dangerous, proposition. Next one. And it is estimated that 60% of cancers will be in LMIC with 5% of resources. So we will have major cancers, major amount of cancer with 5% of our resources. All population, so to the, politics and bureaucrats that they want to do. This is what so this is our this is our ICMA just dialect. There's Google NCBI ICMA or recently. See, this is the statistics is going on for last almost 40 years. I have been in a place for either 270 versus 40. This happens nowhere. In the same country, 7 times difference in the same country, this again says where is the cause of cancer? Cause of cancer is in India. Since cause of cancer is in India, you can find out research. And in female, it's the same thing. Next one, please. It's a place again in Northeast called Bakunpur. Bakunpur is a place in Arunachal Pradesh where the incidence is still high. I told the incidence of cervical cancer has come down, but it has come down all over the country minus Northeast because of their habits and next one. Well, what we achieved, we have achieved phenomenal. Our achievements have been great. These are all what we have achieved. We have a national cancer registry program, the first cancer registry program in the world, and it's a flagship program of ICMA, going on for since 1982. We have a vibrant national cancer registry program, which is called NPCD for control and diabetes, cardiovascular disease, and stroke, because all the others are here. It's a combined program now. Cervical cancer incidence coming down significantly, very good achievement. Many tobacco-related cancers showing downward trend, as shown the results. Stage migration is happening. We used to teach our students a formula of 80-20. 80% come late, out of which 20% is good, and 20% come early, out of which 80% good. That's why people think cancer is a bad disease. 80% of the people don't survive. But today, that is coming down. We are seeing more patients in early stage. And cancer control is really active. You see the amount of money which is available. 120 crores for state cancer issue, 45 crores for tertiary cancer. Professor Saragi said here, the institute, our own institute, our own RCC in Cuttack has received 120 crores. Anyway, it goes by policy. Even a small state like people have also got 120, and they are utilizing. But many states are bought and not utilized. That's the problem. India is a rich country, but some of the Indians are poor. That's our tragedy. And we have to go after it. And most cancers, unfortunately, are memory upset. Some they have to die by cancers. We don't have diabetes, no, we have an India. But we have Tata, slow memory, this method to that. That has to go away. Well, what we have done is, cancer research is a priority now. I will discuss in the last parents, gallbladder, oral, and these are our priorities. Gallbladder, we are the number 2 in the world. Highest incidence of gallbladder cancer is to Chile, 33 per 100,000. And ours in Assam is 70, third in Delhi, 11 per 100,000. Mostly in Delhi, but causes are many that I, people ask why not, why don't we fight cause of cancer? I say the cause of cancer is comfortable without job. So let's go slowly. Well, India, our own data, I was saying, where is our data? Where is our Indian data? The Indian Council of Medical Research, through NCBI, has published our data in published literature, and let me tell you, states for states. The results are absolutely same. Unfortunately, our state score is bigger than their state score. State score is small, more than 4 centimeters, but we have a 15 centimeter. What do you do? That's what. Well, this is the data from our. I will finish very quickly. Well, these are some of the cancers which are showing a next one. Well, the WHO report also shows we have already achieved our target, which was by 2030. Next one, please. These are the survival figures. Again, from back survival. Sorry, I'm touching around back. Well, these are our complications. They are complications on breast, cervix, head and neck, and cervix. All the results are absolutely similar. We have an Indian Cancer Research Consortium now. I'm sure some of you must have applied. Second call of results are out, and there are a lot of money being given for research. These are all again issues we discuss maybe during question and answer session if possible. And if you see, I think Dr. Ghanshyam showed this slide. 14 lakh cancer tests a year, of which 88 lakhs die compared to COVID, where only 6.5 lakhs are that, but less intensive in cancer. Well, challenges in India are plenty, we discussed that, and what I wanted to show is, well, this I definitely wanted to say, this is my baby which I wanted to create. We have created this in India. Now, this is like 300 acres of land. We have got 10,000 crores for this, and we have created an extensive, exclusive cancer research center which is meant for translational. Why? Well, there is an innovated US cancer where there are 4 partners, lives and tells again ICMR and from US side is USNCI. And this is what you saw the photograph of Dr. Harold Ramos, Dr. Ghanshyam showed that, our Prime Minister meeting, and innovated Mr. Obama also. Dr. Tharmas also came to our twice, and we are actively collaborating with them, our scientists and their scientists. We also have a collaboration with trial on Irish drugs. Our first trial on Irish drug, Ayurvedic drug, going to publish very soon. And objectives are again focus on 50% cancer preventable. It is a 17 PI based labs We will produce MD-PhD there, and this is the largest public funded project with 70 acres land. And these are our varieties, which is regularly in India. We have recently got 250 crores from DBT because we have partners that established in the center to do 4 things. First is genomics, early detection research network, EDRN, tissue bank, and animal. And we also participate in the acid program. So population screening, we all know that it has been rolled out in 7 and 8 districts for hypertension, diabetes, and cancers. And we are helping the country to that. We'll answer questions. This is the last slide. Translation research is very critical to care and cure of cancer, essentially to reduce the cancer burden. It should move by direction and where's the part for application personalize the diagnostic recently and help different patient and genetic information to select the optimal treatment. Thank you.
We have questions for something about
We can have brief comments from the chair. For sure, please. That will.
Thank you, sir. I visited NCI and I have seen that fantastic facilities to develop diagnostic research. And
This is actually a guest lecture. Here we will ask some questions from audience. Without question, I don't everybody has listened to everything or nothing. Some questions, some comments, maybe. Yes. Comment from. Sorry, also visited, sir.
I visited NCI when it was being under construction and still fantastic infrastructure being created. So to add, Odisha also stepping very fast. 2015 we had 3 centers, including HPGIC, HRCC, and in the meantime 1 center got closed. So right now we're about 8 centers. So that is essentially because when we met in 2015 an oncology club, there used to be 20 people. And when we meet in oncology club now, there are about 60. So that's the progress we are making. And state is making a very, very rapid progress because our cancer centers are located in such a way. So recently, 5 centers were approved. We have 5 course. Another 5 centers in the pipeline we have in course. It will start with radio centers, medical oncology, diagnostics, community care, and database. Subsequently, we have the surgical information services. And right now, government is very sensitized so far as precision medicine. So we had a very high level meeting in the CM's office where it decided that we should upscale our diagnostic services in terms of pathology, psychology, immunohistochemistry, tumor markers, and more focus on molecular biology. So all the first 4 has been entrusted with the HPGIC and our institution to do it. But we understand that molecular biology is a different ballgame. We are not in a position to start right away. So government is seriously thinking of looking at a PPP partner where this can be done in a private setup and the 100% cost will be borne by them. And tender papers are already ready. We had 2 meetings. We had some identifying what yes we are going to do. Maybe we have given a target and to have a PPP partner so that this problem is solved. And we have been receiving feedback around that for the last 5-7 years. So we decided making a definite stride, but only one problem is our efforts are not backed, which maybe when this infrastructure takes up, research may Because our space centers are right now overborrowed.
She wants to see the.
Once we lessen the cost, once we disperse the facilities across the state, we may catch a little early and the result. Thank you very much.
We must break our cancer road and distribution straight away from today. See, not taking tobacco, there is no cost to save money. So saving on finances and lessening the road, but this is what sort of invented. Yes, sir.
50 years back, we are reading it all by medical college license, which you were not very. I was reading and you were not.
Yes, sir. Yes, sir.
And today, cancer incidence are slowly coming down. By every 7 minutes, one patient was dying of cancer studies. And the incidence has come down because of the persistent effort to get particularly cancer, managing the cancer. But so one of the basic things that I use for vaccination, very few cases are there. Economy is another. That is still at very costly to cover people.
1,500 rupees, example can afford. If nobody can afford, our own applications. 1,500
This has been discussed with the industry, and they have agreed on principle to provide it.
So we have 1,500 rupees per dose on this problem.
If the government provides it, because we have got the rural sector also. Particularly our stocks are the main centers for including the cervical that have several years they used to have this plastic changes. So in that context, I will estimate this test should be prioritized. It is only confined to the estimating of knowledge and some other from no other medical college having that.
What is it done here?
Yes. So
now it's available everywhere.
And for your information, this DNA testing would be increasingly to be done in the medical colleges and in discount part of hospitals.
I don't know what is happening. I think this year we can make it happen.
Right. And one of the there are procedures. One is the transplant procedure. One is the how to select the sample that is very important.
That Dr. Viren
Sir, your suggestions are very, very important. We can discuss, we can implement, we try to implement. So we are running a little bit late, so I request to proceed. So before we proceed, I thank the speakers, our chief guest of this evening, for his fantastic insight of cancer. I said he has shown what India has done in the last 50 years of cancer, from Aizawl to Trivandrum, from Gujarat to Assam. So this is the kind of, I told you, the work Professor has done and has been doing. So this is, let us give a big round of applause for his great work. So before we end the oncology session, it is very important to felicitate our chairperson, Dr. Madhavanandakar.
I request Dr. Banerjee to felicitate our esteemed care panelists, Professor Dr. Ghanshyam Biswas, Medical Director Serum, Professor Institute of Medical Science, Serum, Dr. Soma Surat, Professor and Head, Medical Oncology Institute of Medical Science, Bhubaneswar, Dr. Saurabh Mishra, Associate Professor of Medical Oncology, AIMS.
This is level 1 evidence. Level 1 evidence.
So after a fascinating onco session with four hybrid international panel discussions with MD Anderson Cancer Center and our star medical oncologist team, and then a great DNA lecture by none other than Professor Tikara, we now move into another fascinating session. Here I would like to announce that this term, after precision oncocare, we have given a new term called precision gynae care. And for the first time, I think in India, we are having a panel discussion of precision gynae care. This term precision gynae care has been coined by our great pride of Odisha, Dr. Sujata Kar from Kar Clinic and Hospital Private Limited. I invite Dr. Sujata Kar on stage. Dr. Sujata Kar has the distinction of having the maximum number of gold medals in SCB in her career. And she has been the first to have first IVF baby of Odisha, the first laser hatch baby of Odisha, the first triplet born live in Odisha, and many, many laurels. So IVF, precision, high-end gynecology and fertility has been brought to Odisha and flourished under her dynamic leadership. And I'm happy to announce that she is the Secretary General Designate of the Indian Society of ISPAR, the largest fertility body of the country. 2023, we have the National ISPAR in Bhubaneswar. So, Dr. Sujata is our moderator for this precision gynae care panel. We have very, very important eminent panelists. We have Padma Shri Dr. Kamini Rao, who is a pioneer in the field of assisted reproduction in India. She has specialized in reproductive endocrine gynecology and ovarian physiology and assisted reproductive technology. She has been awarded the Padma Shri for her great work in IVF in the southern part of India, and she now heads as the director of Milan Fertility from Bangalore. We have Dr. Firuza Parikh. Dr. Parikh has been again a pioneer, a game changer in art or assisted reproductive technology at Just Look. She has 42 years plus experience, and she has been many firsts to her credit to get pre-implants in genetic diagnostics to India. She has several gold medals and first ranks in her illustrious career. We have our own Dr. Monu Patnaik on an online mode from Raurkela. He is also a very, very accomplished person at Shanti Memorial Hospital and IVF specialist of repute of 32 years of experience. So we have three panelists, and then we have our own Dr. Mosumi Acharya. May I request Dr. Mosumi Acharya to come on stage? She's again trained in CMC Bangalore, and she heads the Center for Public Management in hospital. So we have two live panels here, and we have three virtual, and then we have our Thermo Fisher technical team from US joining. Thank you.
Thank you very much. Dr. Viren, thank you very much for this opportunity.
Thank you, ma'am.
A very good afternoon, ladies and gentlemen. So we'll try and show that precision gynecology is as important as precision oncology. Of course, precision oncology appears to be more pressing, need and of the hour. And we have attached a lot to our panelists. Can you put the panelists on the top, please?
Yes, ma'am.
Thank you very much, Dr. Kamini Rao, Dr. Firoza Panek, Dr. Monu Patnaik, and Dr. P.C. Mahapatra. I think he is now joining. Thank you so much for joining us. And I would like to make a little correction here. Dr. Monu Patnaik was the first one with the first idea video Orissa. I was the first one for the So welcome all of you. I'll finish up my presentation.
Yeah.
Yeah, and if possible, put the panelists also now.
Both cannot be played together. Both cannot be together.
Screen will be.
Ma'am, that will be on Zoom available.
Fine. So the next slide, and if you could put this on, I'll start with a 55-second video. Put it on.
Genetics, what can it mean? The ability to protect the physical and mental characteristics of every unborn child. In the not too distant future, our DNA will determine everything about us. One drop of blood. Saliva, or a single hair, sex is determined by science. Someone else's. Congratulations, we're at the end. Welcome to Gattaca.
So whether life was created by chance or it was created by a God, the fact remains that it is probably a very, very simple, deep within. It is, and it was written with only 4 chemical molecules. And we should remember that, we all know about Watson and Crick discovered the form of the DNA, but we should also remember Dr. Hattori Purana, who is credited, he's the only person of Indian origin who discovered the genetic code. And that is why we are able to understand the entire genome today. And the point is that the DNA is very, very simple at the base, but it's very, very complex in an individual level. But we have to understand how to read this entire DNA code. We have to use it for any medical benefit. And we, like Dr. Viren said, I think we should also put here. Viren said that we've come a long way. We've come a long way from anatomy of the single cell to molecular medicine, from single genes to the whole genome. We've come a long way. But the future, of course, is about genomics, and the 21st century has to be DNA-based diagnostics. That is what inDNA and people in the translation biological research field aim to do in the future. And it has a lot of application in gynecology, I should say. The major fields that I have taken is about the recurrent pregnancy losses, infertility, prenatal genetic testing, pre-implantation genetic testing. In IVF, we have a lot of role in trying to see the health of the embryo, and in rapid banking, because third-party reproduction is becoming really huge with women delaying pregnancy and needing donor eggs, and even men suffering from a global decline in male fertility. So, I would like to ask my panelists, Dr. Firoza, do you routinely do genetic diagnostic tests in patients with recurrent pregnancy loss? If yes, what tests do you generally advise on a routine? Can we have
First of all, thank you, Sujata, for having me on this program. Unfortunately, I may have to leave a little early in about 15 minutes because I have something else planned. Okay, let's start at the very beginning. We start with something which is simple. We can start with looking at the products of conception by FISH, by karyotyping, by doing microarrays, and we also have the ability to look at the genetic profile of the couple. We need to, of course, do a very simple test first of karyotyping. There are many, many other things, but let's start with the basics, because karyotyping of the parents will pick up many, many abnormalities, which may have been missed if we didn't do it. To give you one example, structural rearrangements can cause multiple miscarriages. We had done a study way back in 2006, I think, where we looked at polymorphic variants in chromosomes. And what we found that if there was a polymorphism, either a lengthening or a shortening of the Y arm, the Y chromosome and chromosome 9, we found that there was a higher incidence of miscarriages and repeated miscarriages. So the karyotyping, although such a simple test, can give us a lot of information, Sujata. Thank you for this question.
Do you believe we should be also looking at POC, product of conception karyotyping? Do you believe
Yes.
in that?
Yes, that is what I said right in the beginning. This is a golden opportunity. So many times we have couples coming to us where they've had multiple miscarriages, but sometimes a medical termination is done, or it has missed the attention to do a karyotyping. There are many things, microarrays can be done, but the basic karyotyping can give us a lot of information, because as you know, one of the major reasons for either early pregnancy loss or pregnancy loss in the first trimester are genetic abnormalities, and these should not be missed.
Right. So as a general guidance, it's the most common genetic test that we all do is a karyotyping of the couple. The POC karyotyping is where we are mostly to satisfy the patient. What about other genetic tests do you do for RPL indication only, like Y chromosome micro deletion? So looking for other causes of RPL, like female factors or anything.
Yeah. So as I told you, we would do a thorough analysis of the karyotyping looking for polymorphisms, and you can catch these polymorphisms particularly in chromosome Y and 9. In fact, now this paper of ours has been cited more than 100 times since we published it in 2006. Of course, there are many other things that can be checked. Y microdeletion may not give us a sense of a problem. Well, it will tell us about one, depending on the microdeletion, whether this person can ever conceive, impregnate his wife or not, or whether if it's a boy, whether this will be carried on to the next generation. But by and large, it will not cause miscarriages. Yes. CFTR, yes, so FMR1 gene is a very important gene, which will be seen in some women with premature ovarian failure or premature ovarian insufficiency. This is many a times in these women, and because of the low AMH, you may have problems in meiosis, non-disjunction, and that could lead to miscarriages.
Right. Apart from regular karyotyping, other specified tests also may be indicated for RPL. But Dr.
I wanted to ask Firuza ma'am, this is Dr. Viren here. I wanted to ask you that from routine karyotyping, now that the technology of microarray has become available and we can go up to 60,000 or 60K array, so we are looking deeper into the resolution of the genome. And is it now being completely followed since the cost has come down and the availability of technology there? And the advantage is here we are not growing the cells, we are just directly taking the gene and hybridizing. So what's your take on that?
Yes. So if the patient is affording, I would certainly ask for a microarray. As you pointed out that the cost has come down, What microarray will do, it will measure the expression of a large number of genes.
Yes.
And because of this DNA hybridization, you will be able to hone in onto the genes with if you are using fluorescent probes. So I think microarray comparative genomic hybridization is the way to go if you have the facilities and if the patient can spare some amount. I think it's a fantastic test. I agree with you completely.
Data. I'm one of the specific, correlation between the genome, and the related to stuff that maybe you have located in our population, because just doing a whole genome as probably help.
Yeah, so that's a very important question. So suppose you are looking at chromosomes. Chromosomes is looking at buildings. So we are looking at buildings. Now we have found that there may be a kind of a inversion line, or there is a translocation, or there is a subtle changes in the chromosomes, which we are looking at a 300 to 500 kb of resolution. But if you go to a 60K array of microarray, we are looking which floor, which room has a glass window broken, and is that glass window allowing the wind to pass, and that building is not efficiently done. So the segregation of chromosomes are not, as Dr. Firuza is pointing out, the segregation effect.
Do we have association?
Yes, we are slowly coming out with association. Now that we have the data available and more data coming, we have been trying to have it.
Right. Can we have the slide, please? And what about sperm DNA fragmentation, Dr. Firuza?
Yeah.
Also is associated with RPL.
Yeah, so not so much with RPL. It is. There is a very important meta-analysis which has been done recently and published. Now, we must make a differentiation between RPL and repeated implantation failure. DNA fragmentation can cause repeated implantation failure. Depends on whether it is a single DNA strand break or a double DNA strand break. Excuse me. If you look at the paper of Casanova in Fertile Sterile 2019, I will urge all of you to read it. There's a very interesting observation where single-stranded DNA can affect some of the parameters of the embryo, but double-stranded DNA breaks will definitely affect even the cleavage and blastulation. So, of course, if it doesn't affect the blastulation, double-stranded DNA breaks, then definitely it may cause aneuploidies, and this in itself can give to repeated implantation failure. There is a paper.
We follow what are common acid, PSPs and SSPs.
Yeah, but we have also published quite papers related to RPL, which is
yes.
related to RPL, right?
In fact, there is a very nice meta-analysis which shows that there is an increase in repeated pregnancy loss also. The mechanism may be different in the two.
Right.
Okay. What is it that is causing change in morphokinetics? Is it just the break, or is it something which is added on, is something that we don't know yet? Everybody knows that it can affect the morphokinetics, it can cause RIF, it can cause RPL, not so much with ICSI. Yes, even now it has been shown that even with ICSI, when we look at the incidence of how much is the DNA breakage, it may translate into abnormal pregnancies and also with miscarriages.
Right. Thank you so much. And now let's come to the next topic, Dr. Infertility. And we discussed with Dr. Kamila. Do you think any advanced genetic tests can be done to find an etiology of premature ovarian insufficiency? Do you do any tests routinely for your patients? Genetic tests?
Question is Dr. Kamini. Question is to Dr. Kamini.
Hello, can you hear me?
Yes, sir.
Yeah, I didn't get your question, Sujata. Can you just repeat that
I
again?
was asking if there are any specific for your cases which you see of premature ovarian insufficiency, do you do any genetic tests on them routinely? If so, which ones do you do?
Well, in fact, some of these are called candidate genes, and it's very difficult to say. These are called either initiator genes, and even if these initiator genes are present, unless you have promoter genes along with it, they will never manifest. So if you look at mothers who have had premature menopause, you will be able to see that you'll have to follow the children as well, whether they will also have premature menopause. The large scale studies which have shown that these do not have full penetrance. However, even the fMRI gene, as Firoza has spoken about, has also shown promise in these areas. However, if one can actually look at AMH at the time of puberty and also at the time of marriage, and then look at the trend maybe 6 months later and see whether the apoptosis is going to be what is the rate of apoptosis, that one, but whether it is going to be only gene related or environment related, I think one has to look at both. Now, if you look at the tropical areas to the Caucasian race, you find there's a lot of difference. So keeping in mind, yes, there is a genetic predilection, but that could not be the only thing to explain the ovarian reserve being low. But there is also an environmental influence, and that I think is something we need to be studying more about.
Right. Ma'am, what about, because making a personalized choice of gonadotropin for IVF, we do that every day, and we don't have too many biomarkers by which we can, exactly pinpoint and take the right doses of gonadotropin or the type of gonadotropin. Do you think we should be doing gonadotropin receptor mutation studies routinely for personalizing ovarian stimulation? Should that be the next step forward? Now that these genetic tests are going to become more easily available.
Correctly.
Am I there? Yeah.
Yeah, there's so much of echo in your voice. Can you speak a little slower?
Yeah. Okay. Gonadotropin receptor mutation studies before, as a routine before starting IVF for a more personalized ovarian stimulation.
Yes.
Do you do it routinely, or do you do it only when your patient fails to respond, or
I think as a routine, I wouldn't use gonadotropins on a personalized basis. However, if the person is actually needing it in terms of whether you're looking at a planned the thing for a young person who has a good reserve and who's only been married for a short time, a person between 25, say 29 with a good reserve, I would rather take only oral ovulogens and rather than gonadotropins. But if she is going in for an IUI, for example, I think controlled ovarian stimulation with gonadotropins and IUI certainly gives better results than just oral ovulogens. So that could be a personalized treatment, if one can call that so. But certainly for IVF, I think one would look into gonadotropins.
Right, and also PCOS being one of the subset of our patients who are really difficult sometimes, and they're at high risk for OHSS. And do you think those category of women also will benefit greatly if we could have genetic tests available to us to check for LH receptor mutations or even NCG mutations and things like that, AMH mutations?
I think if luxury would allow me, I would like to do the genetic mutations and the morphokinetics of the PCOS before stimulating them. But I think if we have the panel ready, yes, because it is such a varied kind of population, the PCOS, it will be worthwhile doing this on the 4 types of PCOS and then go ahead and stimulate them. But if we don't have it, obviously we will have to look at the AFC and the AMH and then stimulate them on the past obstetric history and the past history. But otherwise, yes, if we have the panel, I would certainly like to use it on the PCOS.
So that is what we are hoping for, that people like Viren will be able to give it to us on a nice platform, nice and cheap. Every patient will do IVF, we will be able to do them, do a wide panel of tests, genetic tests that will help us, not totally eradicate OHSS, totally eradicate genuine empty follicle syndrome and things like that. Don't you think so?
Yeah.
Okay. So yeah.
Sujata,
Can I have one of the Thermo Fisher team to comment on it very briefly on this panel? If you have a panel in your pipeline.
Yeah, that would be great.
Our Thermo Fisher friends from US, are you there?
Yeah.
I am there, yes. I cannot turn on my video.
Yes, so you have any panel in the pipeline for, this kind of
A panel which all IVF specialists can advise to their patients, every patient before they start the IVF cycle. And then comfortably give them the correct dosage of gonadotropin and hopefully not have any complications.
We do not have I hope you hear me well. We don't have a panel for this specific indication yet, but we can build targeted resequencing panels for basically any indication, as long as there is a nucleic acid, a genomic basis link to that indication. So that would be my short answer.
Yeah. Okay, so coming back to us that you have.
Sorry, can I just add a little bit onto that? Sorry to interrupt you. Hello? Can I add a little bit onto the panels about infertility?
Yeah, can you briefly tell us something about that? We will tell you something. Yes.
I'm sorry, can you hear me?
Yeah, we can.
Yeah. No, so I just wanted to come back to the infertility panels to add in what Elaine said. In the region, we have worked with a number of sites who have created infertility panels, both for male and female infertility. So as Elaine said, you can create these panels, and these panels are being offered by certain people around, well, within India, as well as around other countries as well. So those panels do exist.
Thank you.
Right.
Sujata, may I add something for PCOS?
Yes, yes, Sabu.
Yes. So for PCOS, yes, there are two things we can use. We can use artificial intelligence, having algorithms. If you have done the gene testing and if you have looked at the AFC, AMH to predict who is going to hyperstimulate and what is a good protocol for stimulation. And at NIRRH, Shrabani has done a lot of work on the genetic profiling and studies of certain genes. And there are 4 important genes and receptors that can be looked at. One is the INSL3 for the production of androgen. Then there is a correlation between insulin-like receptor and AMH. Depending on the dysregulation or the upregulation, one can predict about the OHSS. There's another gene called the PAPAR gene, where you look at the insulin secretion and insulin resistance. And of course, there is PON1, which looks at the reactive oxidative species and looks at oxidation. So yes, one can create these panels for women with PCOS. There are panels which can be implemented, but please remember, many of these panels depend upon ethnicity. We have done a major work looking at AMH of young Indian, and found it very different from. So a panel has to be specific to the ethnicity or the geographical location.
Yeah, absolutely. And I have read Shravani's papers. They are excellent, and we should be, characterizing from the different parts of our country. And she was also wanting to collaborate with the eastern part. I hope something will come out of it. Okay. So, right. So I think I'll ask Dr. Mohan that he has also joined us. Sir, Dr. Mohan.
Yeah.
So what about pre-implantation genetic testing? Do we, in Orissa, are you able to do any of those tests for everybody for your IVF patients or for single gene disorders?
In
pre-implantation genetic testing.
Pre-implantation genetic testing in my place. Though we know there are few things are invasive and non-invasive tests are done. I have personally, I have not done the invasive test. In some centers they have done. So there are conflicting reports, but nowadays people are catching up as they've advanced their technique, and so they are now safe to do PGTA. And sometimes
but we don't, you like Orissa, we don't have too much of a opportunity. Although in Bhubaneswar, inDNA is doing it. We have done a couple of cases. What about Dr. Maushimi? Are you doing or have you got an opportunity?
Ma'am, actually, there are many questions for many patients who are about 35 years and they are really concerned for the antibiotics. PGD should be advised to them. But in Odisha, the main is the embryologist, and second is the availability of the laser, which is required for the procedure. So, I have done 1 or 2 cases maximum in things. But if it would, and the cost factor, all the patients are also not according for the cost. So we are skipping
Cost becomes a major problem. So, Dr. Firuza, Dr. Kamini, you all regularly do PGTI. Dr. Firuza, I'm not sure when you were in Reliance and otherwise also you do a lot of PGTI and you published a lot. Dr. Kamini, Rao ma'am also, they also do a lot of things. So tell me some, like which are the some single gene disorders for which you have had to do PGT?
So there are we have done PGTM for many single gene disorders, for sickle cell, for beta thalassemia, for Leigh syndrome, for Duchenne's muscular dystrophy, for neurofibromatosis, for many other neurological disorders. So we are also now doing haplotyping for beta thal, where we've had successful pregnancies for severe SIB. Sujata, I just want to emphasize that there has to be a lot of coordination for all this between the patient, the IVF doctor, the counselors, the geneticists, the physician, the hematologist. So it has to be a very large team effort. And these are some of the indications where IVF has got married to genetics. And this helps to deselect abnormal embryos and helps to select and create an environment for healthy babies to be born.
Right. Dr. Kamila, do you think, are you happy with the PGTAs that you do, or you would prefer a more, non-invasive? What about the complications? What about mosaicisms and in your routine PGTAs that you do? Dr. Kamila?
Yeah.
Yeah, I was asking you, in your practice, PGTA for aneuploidy.
Yeah.
So, what are the usual indications, and are you happy with the test results? Are you happy with the outcomes? And what about some the major confounder, which is mosaicisms, which are very often reported in PGTs?
See, just like Firoza, we are doing PGS, and particularly for aneuploidy. For single gene disorders, but as Firoza said, you do need this team effort, and of course we have excellent team effort here in Bangalore. So we do a large the same, provided we have the gene probes, and particularly in the area of hemophilia, DMD, provided we have a sibling who's affected and who's alive, we're able to actually test them, and we are able to have something to test against. So the parents' blood and everything is all worked out. But another interesting thing that we have is to have a sibling savior. So what they do is they want to get an embryo, which is a HLA typed embryo. They have tried, and of course, there have been embryos that are HLA typed, but we have not yet put it back into the uterus. But this is something that is actually coming up with the hemophilia at the moment. And the beta thal, of course, we are doing here. And of course, with the Downs, it's a routine. And the elderly ladies, of course, those who are 40 plus, we do it on a routine basis. Now with the ART law that has come in, that doesn't get mentioned in this particular law. So we'll have to wait and see how it's going to be with the regulations, because it doesn't get that mention about age factor alone, other than, of course, where there is pre-existing genetic diseases. But certainly PGT as well as PGA is certainly a boon for getting better results, and of course selecting the right embryo to implant. And certainly the results have improved, particularly in those cases where we have had recurrent pregnancy loss, and where the embryo, where the woman has a Duchenne muscular dystrophy, where she's had recurrent pregnancy loss and these kind of inborn errors of metabolism. So these sort of situations, yes, it does make a difference.
So, ma'am, the new ART law does not allow a routine PGTA?
I didn't get your question.
I thought that's what you said, the new ART law, does it not allow a routine?
Actually mention, yeah, they don't mention about the age factor. They don't say women of advanced age can opt for genetic testing. So that's something that we will have to keep in mind, and whether that gets included in the regulations.
They have not specified, right? So, Dr. Montgomery, do you think how far we are and how quickly we should be having non-invasive embryo biopsy in our regular practice? Because that will make even our life easier. Like Moshimi said, you have to have trained embryologists, the comfort level has to be there. You have to have sufficient number of blastocysts to be able to do a good biopsy. Lately, previous stage biopsy can have do a lot of damage. So a non-invasive embryo biopsy would be the best. So what is your take on it? And then we'll ask where we are.
That is my excuse, Sujata, that because I don't have that facilities around me, so I always bank on non-invasive method. If this thing comes out more standardized, if it is, I am very confident about that, then I will go for non-invasive or micro-invasive PGTA. That is the things which can help me to sort out the people, those who are going for recurrent pregnancy. Because in the setup I am here, I am not have that gadgets and the people around me to help me.
Absolutely.
So is my choice, and once that system is improved and I get confidence on that, I will bank on that.
Dr. Fiducia, you see this as happening?
Yes, culture media and cell-free DNA. These are the things I will be in.
Right. Dr. Fiducia, you see the non-invasive embryo biopsy actually happening in the near future, and then we'll have to get rid of the embryo biopsies.
Sujata, I don't think we'll be able to get rid of the embryo biopsy, the invasive PGT. Well, this can become a supplementation to a PGT program, but one has to have concordance. The lab has to have the ability to do both and have good concordance rates, because without doing PGT, if you straight away go to doing non-invasive PGT, there may be errors in judgment. You have to have a very good IVF setup, you have to get good embryologists, you have to have good geneticists, and culture conditions have to be very good. So although PGT non-invasive will have a place, all who are going to do it have to be experts in PGT.
And we also need to have data and we need to have evidence supporting and correlating. And then I think Dr. Village should tell us how far we are from a non-invasive embryo biopsy.
So a decade back, we were doing amniocentesis and CBS. So then came the era of NIPT, the circulating fetal DNA, and the circulating fetal DNA was kind of regulated now with improved NGS platforms and very low input requirement. We can speak to NIPT quite concurrently. So therefore, if you do the macro features circulating DNA, we can point to the micro, that embryo growing. Okay, when the embryo grows, it also sheds DNA on the media. So if you have enough DNA material, starting material, and then we kind of get sequential, as Madam Day
that we can have a data of the same embryo biopsy as well as the spent media and have a concordance for quite a few number of cases, then we are confident about the invasive as well as the non-invasive, and then slowly move to the non-invasive. Not to forget the invasive is a confirmatory form. So this is how, like if you have NIPT with a trisomy indication, we still go ahead and do an NIPT. Similarly, if you have the indicated, you still go ahead and check that, or maybe you can check later for confirmation when the pregnancy happens. So this is how the scenario is. It is not a 2 + 2 = 4 scenario, but it is definitely a scenario where we can go into 2 + 3, 5 or 6.
So, Sujata?
Sorry, can I jump in here?
Yes, Mike.
Yeah, sorry, can I make a comment here as well? I mean, again, I just want to back up what Dr. Firuza was saying, that you really do need to have a lot of control when you're doing the non-invasive PGT. Okay? But one of the main issues is around maternal cell contamination and contamination of that culture media. So this is easier to spot when you've got maybe a male embryo and and you've got maternal cell contamination. But if you've got a female embryo and you've got maternal cell contamination, you really need to have some form of control to be able to spot that contamination. Okay, so there's a lot of controls that need to come into building that. And I'd also just like to let my colleague Elaine comment, because he's worked quite a lot at Thermo with some of this non-invasive. So Elaine could probably comment a little bit around some of the issues that you face with non-invasive. So Elaine, if you would like to make some comments there.
Yes, thank you, Michael. Indeed, we are developing sets of SNPs, for example, or other genotype markers, just to make sure that there is no, if not maternal contamination, let's say contamination in general, because if there's one thing that we need to change in the laboratory, that is the way the samples are processed. The people should take great care of avoiding any kind of contamination, either from the mother, as we discussed, from community cells, or from the operators wearing specific clothes, for example. So that will need to change in the clinics for sure. But I agree with the precedent speaker. It may be a very, very nice tool as soon as we demonstrate concordance. So we are working with customers to generate more information about that, and hopefully the protocol we already have will be available for everyone. So
thank you so much. So that should be the future. Just let's I think the last point that we can just discuss for a minute, that is ERA, endometrial receptivity assay. I think that also is a very important aspect if we have to personalize our embryo transfer and increase the implantation rate. Dr. Kandida, tell us, do you advise ERA routinely for your patients or in cases of recurrent implantation failure? Do you think it's a useful test? Because there's a lot of debate of ERA now, lot of publications. And there are lots of lobbies who do not believe in ERA, and things like that. Dr. Kamki, what about ERA? Do you believe in it? Do you do it?
You're asking me, Sujata?
Yes, ma'am.
Yeah. Well, I thought, ERA in the beginning, it came with a lot of promise. But apart from the cost factor, what they say is that you have to do it a cycle before, and then about whether receptive and not receptive. And then after that, they say that the immediate next cycle, you've got to sort of mirror image that, and then you have to transfer these embryos. So it is a fairly laborious process. And then you will get it either pre-receptive, receptive, and post-receptive. And then you've got to do it. Now, for example, if you do it, it's only sort of akin to that particular cycle. So what happens is, once you've got that, then you say it is receptive, they actually expect you to go ahead and do it again on the next cycle, keeping in mind the pre-receptivity and the post-receptivity. So if it is post-receptive, you'll have to do it a day earlier. And if it's pre-receptive, a day later. So again, you will have to do the ERA and make sure you've got it as receptive. Now, if you look at the entire process, it's going to be quite a long drawn process and the cost involved. And once you've got that receptivity, then you'll have to go ahead and do the transfer. Now, when I've seen this, one, of course, whether it works or not, well, I've had mixed bag response as far as this is concerned. But then one cannot blame only the ERA, because unless it is only a receptivity issue and all other issues are normal, only then one can say that it could be, a receptivity problem. So my point here is that I have tried it, but I am not totally convinced that ERA could be used as a routine test in recurrent implantation failure. I think unless you have tested the embryo completely, then perhaps one can go in and do an ERA test as a last resort. I don't know what Firoza will have to say. Maybe I'd like her comment on that.
Right. So, Dr. Firoza, the evidence does not, as it is being published slowly, it doesn't seem to be supporting ERA so much. But what about the ethnic and racial differences? Do you think we should have in India, we should have our own panel of the genes that are expressed during the window of implantation, and develop our own panel of ERA genes, receptivity genes, and not depend on just one center which has published and become so hugely commercially popular. What do you think of that?
Yeah. So first of all, Sujata, the ERA is a customized microarray. So what we are doing is we are doing it with a company in Ahmedabad, which has not only looks at 330 genes, but we look at more than 1,000 genes, and these are all customized for our Indian population. So the heat map that is produced for our patients is different for, say, being used elsewhere.
So, that in Ahmedabad?
Yes. We do more than 1,000 genes. It's a customized heat map. So already that is existing. And I am a proponent of ERA, particularly if you use the definition of repeated implantation failure, where there have been 2 failures putting in a euploid embryo as per the recent definition, not just going by putting in a good looking embryo. So if you have a euploid embryo, you failed twice, that's the time to do an ERA. Usually we don't do the ERA twice, because the proponents of ERA, Carlos Simon and all, had earlier said that the reports are valid for about 36 months. Maybe 36 months is stretching it a bit too far, but I would say that it would be all right for at least a year. So we do a lot of ERA even for women with thin endometrium, because as you know, genital TB in our country is very common. And what we see that, and we've done this study, which we are ready to publish, we're just submitting it, we've shown that women who have had or have TB are non-receptive in ERA, and their pathways for expression of implantation, there are many, many pathways, the MAPK pathway being the most important one. This gets upregulated. So if the inflammatory molecular pathways are upregulated, and if you have an ERA which is non-receptive, then you have to counsel the couple and say that perhaps even with doing a customized ERA transfer, a personalized embryo transfer, it may not work, because the progesterone receptors are down-regulated. The progesterone receptors are eaten away by the tuberculosis bacterium. So in our hands, ERA, particularly for certain situations, works very well.
Thank you so much. So I think we just have the last word for tonight about gamete banking. So third-party reproduction is becoming very important, and there's plenty of gamete banks opening up. The new ART coming in, the oocyte and the sperm banking has to become much more strict, and we need those gamete banking. And at this time, we are only so both Moshmi and Dr. So we are just doing some minimal tests on the donor, which is the blood group and maybe some infections like HIV, HPS, HPV, etc. Do you not think a detailed genetic profiling of the donors would be a great idea once we have that platform available? What is your take on it? Moshini, you can answer and then we can see.
Ma'am, this is the right question because we are all like PGT, PGS, and also patients we are definitely giving donor option. So basically, the first concern of any patient who is receiving a donor is whether you have screen for defects, whether there is any hereditary defects we expect. We don't have an answer to it. Just doing a normal karyotype may not. So it is definitely when the rising cases of third-party programs are coming, it's a definitely say the gamete banks should be equally professional qualified to do all the screening tests to rule out the common genetic disorders. Single gene disorders, common disorders may not be the complete.
For example, at least in Orissa, thalassemia, and minimum
which are which may be life threatening to the
which are doable and maybe that particular.
It should be done and ethically it should be done, but the recent practices like only the donor with maybe a karyotype and even not a genetic counseling to the donor person. The sperm banks, I think they are a bit more professional, but the oocyte is still not streamlined or they are not screening the patient. So it's real concern.
Right. So I think we can just wind up by saying what are the chances that we will be able to do such genetic tests? What will be the cost involved? Is it possible? Like once it's supposing to start a sperm bank, how far can you test a donor? And how expensive will it be? Is it practical to do it? Because at this time the sperm banks are very few to do this.
Yeah. So my take here is, of course, with the technology, we were getting a blood test done with your routine biochemistry, CBC, and hormones. But now we are going into high-end profiling in the blood. So when the testing paradigm or the testing horizon is becoming molecular, is going into the DNA. Why not profiling the donors with the available technology of the at least the standard hereditary disorders? So that we can confidently counsel the recipients or the couple who is receiving the gametes that yes, these gametes are free of the standard disorders which you have in mind, like they're non-thalassemia, they don't have a single genetic disorders, or the donor do not have a family history, known family history of any genetic disorder screen, and it's a apparently genetically healthy gamete. But later on, what happened, that's not in our hand, but at least we can confidently counsel, as man was pointing out. So we have some points of confidently counseling rather than just telling that we have done routine blood test, infection we have ruled out, and this is the age of the donor. No, we can also tell that genetically the donors are not.
Right. So I think we had a very informative and fruitful discussion on the main points or the main aspects of precision gynae care genomics in gynecology. And we covered quite a lot. Thanks a lot to all the panelists for giving their valuable inputs. Thank you, Moshini. Thank you, Dr. Mohammed, Dr. Kambi Rao. Firuza was busy, but thank you so much for being there, Dr. Firuza, for a long time. Thank you, Richardson, Elaine, for being there and giving your inputs. I think that's about all.
I just want to add one 30-second scientist. Ma'am wanted to say.
Dr. Kambi.
Ma'am, please go ahead. Kamini, ma'am, please go ahead. Yes.
Yeah, I just like to say that in memory of Dr. B.A. Chakravarti, I'd like to say my pranams because this will be the first time in East that I'm coming on the screen. So my big pranams to Dr. B.N. Chakravarti, the doyen of IVF. And I suppose this has been perhaps the first scientific program that I've been on. And I'm sure all of us remember him and would dedicate at least my part of this program to the great legend, Dr. BNC.
In continuation, this is called the blessing of Professor B.A. Chakravarti. Because I was just going to tell that this session we want to dedicate to none other than Professor B.N. Chakraborty, who has been a father figure in IVF in this country and an institution in his actually, I have seen such an enthusiastic, curious man at the age of 94 talking to me at midnight PM. Have you read this paper? This is exciting. This is very good. So this is the kind of inspiration we used to draw from this man. So it's really great that we are taking his legacy forward in marrying the science with the practice. And here, see, I would like to just mention that Dr. Firoza Parekh was busy going somewhere, but the scientist in her actually made her sit back and listen and
Exactly.
So this is the passion we all carry beyond our profession, beyond our practice, that the science and the curiosity will keep us moving, and that's why we are all here. Thank you so much.
Thank you so much. Thank you so much, sir.
Thank you so much, all the respected delegates, for your valuable and enlightening words. Now, I would like to invite Dr. Arun Chochraj, one of the founding members and director of inDNA Life Sciences, to come on stage and felicitate our respected panelists.
A very good evening. I would just request Sujata ma'am and Moshika Chakran to come on the dais.
Dr. Sujata Garg. Dr. Roshni Acharya. Now I would request Anand to take over the next part of the session. Thank you.
A very good evening to the August gathering here on the occasion of World DNA Day. We as inDNA celebrate this day with great pride and passion, as DNA, as a wonder-born molecule, allows us to know exactly and see who we are and how we are doing. And this platform of inDNA was created with the vision of translating bench research or science of the laboratory for directly for the benefit of the patients. For inDNA, for us, patients come first. And today, we are privileged to have some of the beneficiaries of inDNA to be with us. So to honor them, I request Manisha to kindly call out the names and would like to just honor and felicitate some of our beneficiaries.
I'd like to call to see Chandrananda Garg. Please kindly come on the stage. Next, Mr. Vincent Lenga. Vincent Lenga.
Hey, these are certified organizations. R&D and training are an integral part of our activities. So we have been offering hands-on training with the life sciences and biotechnology students on that-based DNA technology at our inDNA labs. To commemorate the World DNA Day, we have organized the poster competition where students from Odisha had participated, and we would like to now announce the winners of the poster competition. Manisha, can we have the winners' name, please?
So the second honors prize goes to P.K. Kanchan from Serum University.
Posters of the winners are lined on the hallway. So please make time to see if we have displayed if they are the winners.
First runners-up prize goes to Sudhireka Mishra from the University. And the first prize goes to Svetarani Das from Serum University. Very much congratulations to you. Thank you.
Now I would like to propose a vote of thanks. It is my honor and privilege to propose the vote of thanks for the 7th World DNA Day that we have celebrated today, and I can see that it has been a grand success. I want to start with Professor G.K. Raj for blessing us, sir. Your presence here is a blessing for us. And we express our heartfelt gratitude for you to inaugurate our EVA position clinic and also accepting your request for the thank you very much. I will now thank Dr. Lalahani sir in absentia for inaugurating our GC testing facility today and also for being our guest of honor. Next, I would like to thank the physician care panelists, and we had a stellar participant from India as well as abroad. So to start with, I want to thank the ILON team who will be the collaborators with the inDNA, and we have a very bright future together because there are a lot of dreams to be unfolded. So I want to thank Dr. Sandurai Mani, Dr. Vivek Subaya, Seventhi Nimai, Dr. Seventhi Nimai in absentia. Also, I would like to thank our very own Dr. Ghanshyam Viswas. Sir, thank you for moderating and organizing a very interesting panel discussion in the precision onco care. Those of us who were non-technical would also understand that yes, things are moving in a positive way. Thank you for that, sir. I also want to thank Dr. Swami Panda, Professor Swami Panda, Dr. Saurabh Mishra, and Dr. Manish Singhal, who joined us from Delhi. I now want to thank the precision gynae care panelists. To start with Dr. Sujata Kar, who moderated an enlightening session. It was so interesting for everyone. I believe everyone enjoyed it and everyone was engrossed in the discussion. I want to thank Padma Shri Dr. Kamini Rao, Dr. Firuza Parikh, Dr. Monu Patnaik, Dr. P.C. Mahapatra, his blessings are always with us in absentia, and also Dr. Moshmi Acharya, who joined us in the panel discussion. Thank you for being here and thank you for supporting inDNA all the time. Today we are really overwhelmed by the presence of so many of our clinician partners who have been with us from thick and thin. And it gives me a lot of happiness because it seems like a dream come true. for us that you all have blessed this occasion and you have given your precious time. I know doctors are extremely busy. You have missed your clinics. It's a Monday evening. You have missed your appointments and thought of being with us here this evening. So thank you very much for spending this evening with us. I would now like to thank our sponsor, one of the sponsoring partners, Thermocare. We saw some of the panelists there who joined from the US. Also our media partner, the students who joined in, and we are blessed with the presence of some of the senior doctors in Bhubaneswar and Cuttack who joined in spite of their busy schedule, and it gives us pride to felicitate two of the doctors that I can see, Dr. Kripashindu Panda and Dr. Siddharth Gaurav. So, if you would join us on stage, we would please like to felicitate. Please, sir. Dr. Siddharth, sir.
भेजो ना अगर है तो व्हाट्सएप में सर बोल रहा भी मांग रहे हैं मेरे से.
Thank you, God bless.
So I want to thank the inDNA family, each and every member of inDNA family for tirelessly working for organizing such a stellar World DNA Day program. Thank you everyone. Thank you very much. I just want to leave all of you to the video from our technology partner, Thermo Fisher, because Thermo Fisher provides the maximum instrumentation facility at our university. So with that, I also declare the dinner is open, so please approach for dinner. Thank you so much.
Sponsored for. And in the Survey, serving as